Hey everyone, today we're diving deep into one of the coolest, most essential processes in all of biology. We're talking about the Great Genetic Shuffle, otherwise known as meiosis. It's this absolutely brilliant mechanism that makes sexual reproduction possible, and we're going to break down exactly how this incredible cellular dance works.
So let's kick things off with a question that seems, a little absurd on the surface, but it gets right to the heart of why meiosis is so important, right? We all get our genes from two parents. So why don't we just end up with double the amount of genetic stuff?
I mean, if you just did this simple math, the first kid would have 92 chromosomes. That's double what's normal for a human.
And it would just keep going, right? The next generation? Well, they'd be up to 184 chromosomes.
Yeah, you can see exactly where this is headed. In just a few generations, you'd have total and utter genetic chaos. But obviously, that's not how life works. Because life, well, it came up with an incredibly smart solution.
And that solution, the hero of our story today, is meiosis. This is the special kind of cell division that stops that genetic pileup and makes sure everything stays stable from one generation to the next, while also, and this is key, adding a little bit of variety into the mix.
So what is it exactly? Well, meiosis starts with one of your regular body cells. We call these diploid because they have two sets of chromosomes, so 46 in total for us humans. Then, it puts that cell through not one, but two rounds of division. And what pops out at the end? 4 very special, very unique cells that we call haploid, each with just a single set of 23 chromosomes. And you know these cells as gametes, sperm and eggs. You can think of meiosis as having two main jobs, two core missions. First, stability. It cuts the chromosome number in half in the sperm and egg so that when they get together, the new baby has the right number, 46. Mission accomplished. But second, and just as important, is variation. Meiosis doesn't just make clones, it actively shuffles the genetic deck, which is absolutely vital for things like adaptation and evolution.
All right, so the whole process happens in 2 big stages, kind of like a 2-act play. We're going to start with Act 1, meiosis I. This is often called the reduction division because it's where the chromosome number gets cut in half. It's also where all the really cool genetic shuffling magic happens.
Meiosis I unfolds over 4 phases. You've got prophase I, that's the big genetic shuffle. Then metaphase I, the random lineup at the center. After that is anaphase I, where the pairs get pulled apart. And finally, telophase I, where we get our first 2 cells.
Now, the real main event of meiosis I happens right at the start, in prophase I. Your homologous chromosomes, that's the matching chromosome you got from your mom and the one you got from your dad, they find each other inside the cell. And then they do something incredible called crossing over. They literally swap pieces of their DNA. Think about it like taking 2 different decks of cards, one red and one blue, and shuffling them together so perfectly that some red cards end up in the blue deck and vice versa. That's how we get brand new, totally unique combinations of genes.
And the quest for variety doesn't even stop there. In the next phase, metaphase I, these freshly shuffled chromosome pairs line up in the middle of the cell. But, and this is so cool, how they line up is completely random. Which side the mom chromosome is on versus the dad chromosome is a total coin flip for every single pair. We call this independent assortment, and it adds another huge layer of mixing. For us humans, this step alone creates over 8 million possible combinations. This is wild.
So, at the end of meiosis I, our original cell has now split into 2. And here's the crucial part. We now have 2 haploid cells. We've successfully cut the chromosome number in half, from 46 down to 23. But we're not quite done yet. There's still one more step. And that brings us to Act 2, meiosis II. Now, if meiosis I was the complex, creative, shuffling part of the show, meiosis II is much more straightforward. It's really just a second, simpler division to finish the job. In fact, meiosis II is a lot like another kind of cell division called mitosis. That's the one our bodies use all the time for growth and repair. The goal here is super simple. Each of those 23 chromosomes still has an identical twin attached to it. We call them sister chromatids. Meiosis II is all about just separating those twins. The phases even have the same names: prophase, metaphase, anaphase, and telophase. But this time, the chromosomes line up one by one, and in anaphase II, those sister chromatids are finally pulled apart. So what's the result? Well, our 2 cells from meiosis I divide again, and voila, we have our grand total of 4 unique haploid cells.
Okay, so now that we've gone through the whole journey, it's super helpful to zoom out and put meiosis right next to its cousin, mitosis.
Seeing them side by side really makes it clear why each one is so special and so different.
Looking at this chart, the differences just pop right out at you. Mitosis is for growth and repair. Its whole purpose is to make identical copies, perfect clones. So, one division, two identical diploid cells, and zero variation. Meiosis, on the other hand, is built for sexual reproduction. Its entire goal is to create variety. So it uses two divisions to make four unique haploid cells, all thanks to that amazing genetic shuffling we talked about.
But there's one last fascinating difference in how this all turns out. In males, all four of the cells made by meiosis go on to become viable sperm. But in females, it's a different story. The resources, like the cytoplasm and all the good stuff, are divided unevenly. One cell gets almost everything and becomes the egg. The other three, called polar bodies, are tiny and just kind of fade away. It's a strategy to concentrate all the nutrients needed for an embryo into one single powerful cell. Pretty smart, right?
Now, meiosis is an incredibly precise and tightly controlled process, but like any complex piece of biological machinery, sometimes errors can happen. And when they do, the consequences can be pretty significant. The most common type of error is called non-disjunction. It sounds complicated, but the idea is actually simple. It's just when the chromosomes fail to separate correctly. It's like they get stuck together during anaphase 1 or anaphase 2. The result is that one cell gets an extra chromosome and another cell is missing one.
This leads to a condition called aneuploidy, which just means having the wrong number of chromosomes in your cells. A very well-known example of this is trisomy 21, where a person has 3 copies of chromosome 21 instead of the usual 2. This results in Down syndrome. It's a really powerful reminder of just how critical the precision of this whole process really is.
All right, we've been on quite a journey, huh? From that initial math problem to this elegant cellular solution, and even its potential problems. So what's the big picture here? What's the grand takeaway from all of this? I think this quote just nails it. Meiosis isn't just about splitting a cell in two. It's fundamentally a process of creation. It manages to do two things at once. It creates the genetic stability that lets life continue from one generation to the next, and it creates the genetic variation that fuels all of evolution. It's an incredible balancing act.
And that leaves us with one final thought to chew on. This beautiful biological dance perfectly balances stability with creativity, order with randomness. So what do you think this deep-seated principle, this idea embedded in our very cells, tells us about the fundamental strategy of life itself?